today we have Ethan. Ethan has been working with Andrew on layer-fMRI, motor learning. Ethan is in the group of Liu Cohen in NINDS. And actually, this meeting was planned from Andrew. That's why I asked you. Sorry for the.
Oh,
yeah.
yeah.
And the title is Layer-Specific Dynamics in Primary Motor Cortex Related to. I cannot see it. Sorry. There's the rapid skill learning in humans. Yeah. Thanks, Ethan.
And should I?
You can.
Okay, so thanks for the invitation, Andrew and Renzo. So today I'm going to be discussing a relatively recent project where we've completed data collection on that we ran on the NMRAF-7T scanner using a sequence that Renzo kindly developed for us at the beginning of the pandemic, I think. Good time for development work. So I'm going to start with some previous work we've done in the MEG to kind of orient you to our interests in the lab and studying skill learning and how we go about doing that from the behavioral standpoint, and then some of the previous findings we've had with MEG, and then I'll move into the more recent VASO results. And so Andrew was, very involved in this study, both from the development and data collection standpoint. And I also want to acknowledge a former postdoc fellow in our group, Roberto Salamanca Garon, who recently left us to move on to greener pastures. And, both of them were instrumental in the VASO analysis work that I'm showing here today. So human skill learning involves forming new memories that occur through a binding process that hierarchically links simple action elements such as a single key press on a piano to construct more complex spatiotemporal sequences, such as when you're playing a piece of music. And we're interested in investigating the brain mechanisms that are driving this binding process and consolidating these memories into temporally precise skills. And it's repeatedly been shown that when you interleave rest breaks with short periods of practice, you actually improve the skill acquisition and memory consolidation process. And so the spacing effect could, in fact, be a form of rapid consolidation, which evolves during waking rest. And the way that we study skill learning in the lab is we use a task that's very similar to this piano example, where we essentially should display a kind of symbolic key press sequence on the screen, so similar to a piece of music, sheet music, and then the participant learns to basically play this sequence in a repeated manner as quickly and as accurately as possible. And so the goal for them is to basically increase their speed and accuracy, or maintain their accuracy while increasing their speed after several rounds of practice. And traditionally, this has been studied in laboratories with respect to sleep consolidation. And so essentially, participants would train on this sequence during an initial session over several minutes. And this period, it would be referred to as online skill learning. So it's performance gains that occur while there's actual practice occurring. And you see, typically you see this initial rapid gain in performance that's followed, that quickly asymptotes, after the first few trials, and then it's followed by these like slower linear gains over the remainder of the training session. And what's interesting is that, if there's an intervening period of sleep or even a long passage of time where there's no additional practice, when you bring the subjects back to retest them, you see this like large increase in performance, which is referred to as offline learning gain. Okay, so something is happening to consolidate the memory during this period that actually leads to further performance improvements. And what's happening is that these sequence skill is actually exhibiting this binding property like very early in the training session during this early learning period. And so if you see that the overall sequence speed is increasing here, if you look at the actual key press pair transition composition, that's the components of the sequence, what's happening is that this pattern of key press transitions is becoming much more regular and stabilized. The patterns are stabilizing, according to this example from an earlier data set we have, by trial 6, and it remains stable over time. And what's happening is that there's an overall kind of improvement in the sequence speed, but the underlying kind of structural composition is remaining stable. And we actually have a new data where we've actually recorded kinematics, and it shows a very similar effect from the kinematic standpoint. Whereas in the first few trials, the variance in the kinematic data is explained by individuated finger movements, and by trial 6, a majority of the variance is actually explained by synchronized movement synergies across all 4 digits. And so one question that we asked is, when does this online skill learning actually occur? And so if you kind of look at the skill and performance gains over like finer temporal resolution, you can estimate the instantaneous performance of the skill within a single trial, and then you can look at the changes occurring within a trial, like during the actual practice, and we refer to that as micro online learning. And then you can look at changes in performance that happen immediately after these intervening periods of rest, and we term that those changes as micro offline learning. And what we found in an initial MEG study that we conducted next door was that essentially almost all of these early learning gains you see here, which explains 95% of the total learning on the first training session, are explained by these micro offline gains. So essentially what's happening is that every time you practice for a 10-second period, and then you take a 10-second rest, when you come back and practice again, you see a big jump in performance. And these jumps in performance are explaining all of the learning that's happening. And we were able to replicate this in a crowdsourced online study, because this task deploys very well to that setting, with several hundred participants. We found the same effect. And in that study, we also ran additional experiments, trying to control experiments, trying to understand this effect more. And so we found that these micro offline gains are not explained by fatigue. So when you substantially shorten the practice trial so that you're not seeing any drop off in performance during the actual practice, you still see these micro offline gains. And then also, if you introduce interfering sequences at different points throughout this rest period, you also see that these rest periods are important for memory stabilization, indicating that these gains are likely some form of rapid consolidation. And so we became interested in whether, and obviously in the neural mechanisms underlying this effect. And one thing we decided to explore was the role of neural replay. So replay is the fast, temporally compressed reactivation of neural activity patterns during waking, rest, or sleep that represent action sequences or trajectories. And these replay events can represent the action sequences either in forward or reverse directions. And most of the initial work with replay was done with respect to navigation, spatial navigation, memory systems, and rodents. And more recently, it's been extended to more reinforcement learning and declarative memory sequences in humans using MEG. And most of this work's been done with Ray Dolan and Tim Barron's groups at UCL. And so our hypothesis with respect to replay and its relationship to micro offline gains was that neural replay, the skill sequence, should be prominent during inter-practice rest if there's a possible underlying mechanism here. The replay density should be modulated by practice. Any replay that we see should be selective for the practice sequence structure if it's reinforcing the memory of the skill, and the replay density during the short inter-practice rest periods should predict the observed micro offline gains. And so this was the study design for our MEG study. So it's pretty simple. Subjects receive task instructions on the sequence they would be performing here, followed by pre-training resting state scan. And then they performed this sequence over 36 trials. Each trial was 10 seconds long, and it was interleaved with 10-second rest breaks. So we refer to that as inter-practice rest. And then at the end of the training session, we did a subsequent post-training resting state scan. And the way that we investigate replay using MEG is first we use our practice data to basically we source localize our practice data, and then we average it using a functional area parcellation approach. So essentially, we're using template defined ROIs to average our voxel activity, and then we fit. In this case, we fit SVM decoders for each individual key press to get an estimate of the brain representation of the individual key press states. And then we evaluate the decoders on held out data, and you can see we get pretty good performance with our decoders. And then what we do is we apply our decoders kind of in a temporally sequenced manner using different delays. And we apply that to our rest data. So the data collected either in the pre-training or post-training resting state scans or in these short inter-practice rest periods. And then we can basically perform statistical testing to determine when we see a statistically significant replay reactivation event. And we can do this for different sequences. We can do this for forward and reverse directions of the training sequence. And so what we found in the MEG study is that indeed we do see a large increase in replay density that's induced by practice, and this is forward replay of the train sequence that I'm showing here, and that it's prominent during this early learning period. So we're over the first 12 trials of the training session. And when we look at the kind of source distribution underlying these replay events, so essentially here what I'm showing is we've epoched our rest data around these replay events, and then this is just principal component analysis showing the regions that are showing covariant activity during these replay window. What we see is a network that includes the hippocampus here, surrounding perihippocampal regions such as entorhinal cortex, and other mediotemporal regions. And then importantly, for the vaso work that I'm about to show, we see a strong involvement of the contralateral sensory motor cortex, including M1. We found that the replay is definitely selected for the practice sequence. So both forward and reverse replay of the practice sequence showed a significant elevation immediately that was induced by practice, whereas replay of a control sequence that doesn't share any kind of structural similarities with our training sequence. So there's no shared ordinal key press locations and no shared transitions. We found no increase for that. And then if we relax our criteria slightly to allow either one shared ordinal key press location or one shared transition, we also see no change in replay events for those sequences. And then finally, to wrap up this part of the introduction, we did see a behavioral effect. So subjects that showed greater replay density during these interpractice rest periods for either forward or reverse directions of the train sequence. These were the subjects that showed greater micro-offline gains, and that this relationship did not occur for replay of the control sequence. So to summarize, we did find that neural replay during early skill learning was highly prominent within the same interpractice rest periods within which rapid memory consolidation occurs. The observed replay was temporally compressed. So the replay events were about 50 milliseconds in duration, whereas the single iteration of the skill after learning was about 1 second long. The replay was spatially represented across a distributed network that included the hippocampus, mediotemporal, and contralateral sensory motor cortex. The density increased sharply during training, and these increases were selected for the practice sequence. And finally, this replay density was highly correlated with the rapid skill consolidation we observed. And so the main question that we wanted to address with our VASO study was, is rapid skill learning and consolidation associated with reorganized input-output dynamics in M1? And so, this was really motivated by, the work that Andrew and Renzo published previously. So Andrew basically showed that this nice dissociation between motor imagery and execution of finger tapping movements across superficial or across vaso responses across superficial and deep layers in M1. And so the idea is that these superficial layers are reflecting basically cortical input or thalamocortical input, and then the deep layers are reflecting changes in corticospinal output. And so we were interested to understand what was happening with respect to these factors during the early skill learning process. And so this was the design for our VASO study. So learning studies are a little bit tricky in that you're essentially trying to capture some very dynamic state that's changing very rapidly over time, and at the same time that you're trying to be able to estimate that process, it's pretty noisy, and you don't want to kind of do anything that will interfere with that process initially. So you can't do kind of typical things like counterbalancing conditions that you would typically see in a MRI study, especially a block design study. The other challenge is that we know from our MEG work that there's some important processes that are occurring during these rest periods that we want to be able to evaluate. And so typically this these rest periods here shown in white would be used as a contrast for your task period, but we wanted to avoid that here. So we have our two localizer blocks that were evaluated at these 2 time points, and so this is actually a general localizer scan that gives us a mass that identifies a single representation for all 4 finger digits involved in the task. So that differs slightly from some of the work that Renzo did previously. And then, of course, we're concerned with the fact that if we have any kind of confounding factors that are influencing the VASA response, in this case, they're going to be correlated with learning. So as I showed you earlier, these learning curves are kind of single exponential curves. So you get a monotonic increase in performance over time, right? So we have, a VASA response that's being affected by, just the passage of time, or that's, correlated in some way with the tapping speed. We wouldn't be able to basically parcel that out of our learning effect, because all 3 factors would be correlated. So we introduced this random sequence block here, where essentially on each trial the subjects are performing a different sequence, and their performance on these individual sequences are highly determined by the structure of the sequence. So some are easier, some are harder, right? And in this way, we're able to basically uncorrelate time and speed, or that's the goal anyway, and also have a condition where there's no learning that's occurring over trial. And the idea was that we could then, if time and speed are indeed important factors in predicting the VASA response over trial, that we would then be able to basically predict those effects using regression model fits to this data set, and just apply them here because everything would be within the subject.
So I have a quick question. Sorry, if I could interrupt briefly. Do you worry about like meta learning with the random sequence block, like kind of just learning in general, how to, taking on new sequences?
Right. So you do see the meta learning that you do see is that all of the performance on these sequences in the second block are going to be slightly elevated compared to the performance on this initial trial here. But the idea was that we have enough data from testing different sequences in our kind of online crowdsourcing work that we can estimate, but we can estimate which sequence structure should give us like a good range, because our primary aim is that we're able to kind of match the full range of performance speeds across this first block. You are right that there is going to be this kind of element in the second block where they have experience with performing this task, right? And so definitely what will generalize are things like, they know they have more experience with this symbolic representation of the individual key press actions, right? So they may not be as confident in the initial trials here with those relationships, but they're very confident later on. But that's the primary thing that we see generalizing. Yeah. And so this is the behavioral the data on the training sequence block from our VASO study. And so, I just want to point out that, we see very similar findings to what we saw in our previous MEG study, as well as the crowdsourcing work that we've done. And so what you see is this rapid increase in performance on the sequence over the first few trials, and it reaches 95% of peak performance by trial 12. So in the previous work in the MEG, it was trial 11, so very similar finding, and that all of the learning that's shown here is explained by these micro offline gains. So it's explained by these, jumps in performance after the intervening rest period. And this is to show that, our random sequence block behavior essentially was, what we predicted, and that we see a range of performance speeds that largely cover the range that we see in the training sequence block. So it ranges here from right around 2 key presses per second to just above 3.5. And you see what you're only missing this initial point here outside of that range, really. The other important thing is that you don't see kind of this monotonic increase over time. So there's no real kind of over trial learning effect here. And when you rank the sequence speeds across individual subjects, so this would be the median rank here, and then these error bars actually show the full range of rankings. What you see is that these first 3 sequences, they're typically performed faster by subjects because they have these kind of like 4 consecutive key press progressions. So 1, 2, 3, 4 here, 4, 3, 2, 1 here. And because they're doing this repetitive, you have to think of these as circular sequences. And this third one has another 1, 2, 3, 4. And so it's these types of structures that are largely driving, how hard or easy these individual sequences are, as well as kind of relations. There is some relationship, overlapping relationship between, kind of doublets and triplets here versus the train sequence. And so if there's more shared structure in that respect, you do also see better performance here. But the main take-home point here is that we essentially accomplished our aims for this block, as you can see in the behavior. And so this is the VASO and BO localizer responses that were fit using the localizer data. And so on the left here, you see the group average. So this is actually oriented opposite, I think, to how it's typically shown, where typically you're seeing superior on the left, deep on the right. These green bars here are just showing where there's significant difference between VASO and BO. So that's not important. But the main thing is that we see this clear kind of double peak profile that we're looking for in the VASO localizer data. And you can see that it's prominent across almost all subjects. And actually this subject was held out of the analysis because of they didn't comply with the task constraints. So and so thinking about our previous findings with MEG and Andrew's findings in relation to the motor imagery work that he did, our prediction was that that during the rest periods we would see a we would see a relative increase in vasor response in the superficial layers that would be modified by learning, and that this would not be the case in deep layers. However, it is important to also consider this repetition suppression effect that Andrew found in the superficial layers as well. And since in our training block, they're repeating the same sequence over trials, we have to consider that this repetition suppression effect may be kind of impacting the results as well. So I'm going to show the vaso data from our random sequence block first. And so this is actually showing all the data for individual subjects over the full 20 trials for the random sequence block. And the white vertical white columns indicate the practice trials. When it switches to the gray shaded columns, that indicates the inter-practice rest. The blue are the deep layers, so the vasor response for the deep layers, and the red is for the superficial layers. And the main thing that we're seeing here is that we don't see any temporal trends in the data. So this suggests that there aren't any effects that are caused by kind of the passage of time or time engaged in the task, as well as there don't appear to be any effects related to tapping speed, because I showed you before that that's varying quite a bit from trial to trial. What we're mainly seeing here is that in the for the deep layers, when you transition from practice to rest, you see a decrease in the vasor response. And when you transition from practice to rest for the superficial layers, you see an increase in the vasor response for this random sequence block. And just to kind of point out, so this is a scatter showing the vasor response, and everything's mean centered here for both deep and superficial layers. So these are all individual data points and showing the relationship with the correct sequence speed. And so you don't see any real speed effect on the vasor response. And this is the case for both the practice periods, and then if you're looking at the relationship between the vasor response and the following inter-practice rest period with the preceding speed, you don't see any relationship either.
Is that expected? Like it helps your case with, so you can separate learning from speed, but if you move your fingers more, wouldn't you expect that kind of the output neurons at least need to?
Yeah, so this was like we had designed the study with this expectation, and yeah, we didn't find it here. So I do know of like bold literature that shows a scaling of bold with force production. But I'm not too sure about any previous findings showing a relationship with speed. But we did expect to see a relationship.
It's kind of
and when you run the regression, the explained variance is super low, like less than 4%.
It's not only speed that doesn't matter, but also kind of complexity in the response.
Right.
Again, right.
Right.
Interesting. Okay.
And so when we look at the vasor response over the full main sequence task block, we also don't see any kind of temporal over trial trends in the data really. But one difference from the random sequence block is that we do see that for both the deep and superficial layers, we see a decrease in vasoresponse during rest relative to the practice period. And then so these are essentially direct comparisons between the training and random block data. So the top row is the superficial layer data, and the bottom row is the deep layer data. And then on the left column, we have practice. On the right column, we have rest. And so what you're seeing is that during practice, for the main training block data, you're seeing like an increase in vasoresponse, slight increase relative to the superficial layer data during practice, during the random block, and that during rest, there's a decrease relative to random. And then for the deep layers, there's no difference between training and random. So it does seem that, that we are seeing some difference related to the, training versus random block effects that are, that's isolated to the superficial layers only.
What are the units of the vasoresponse?
Sorry, what are the units?
Yeah, like like what is the vasoresponse? What type of measurement is it?
Yeah, so what are it's
It's the signal change.
yeah, the signal change, it's and what are the units again? It's
What is
it's
it
the same units as Andrew's paper, so it's
percent signal change, yeah. Like
Percent signal change, okay.
yeah, percent signal change. And so to summarize, we didn't see any apparent temporal trends in laminar vasoresponses over early skill learning during practice or inter-practice rest. Over the training block, vasoresponses for both superficial and deep layers decrease when transitioning from practice to rest. While over the random block, the same decrease is seen for deep layers, but an increase in vasoresponse during inter-practice rest is observed for superficial layers. And then finally, vasoresponses in superficial layers over the training block are slightly enhanced during practice and attenuated during inter-practice rest relative to responses over the random sequence block. And so one thing, I wanted to discuss next was, now that we've run the study, and have seen the results, there's a few ideas that we have about maybe why we're not seeing more robust effects related to learning. So one idea is related to the some work from Jorn Diederichsen's lab, where they've done lots of bold fMRI work with sequence learning. Typically, they're using longer sequences and they're training subjects on actual sets of sequences over several weeks, and then looking at changes in bold response patterns, so multivoxel response patterns in many cases over these weeks. And then what they're finding in their work is that a lot of the kind of sequence learning representations that are emerging following training are represented by changes in these multivoxel patterns as opposed to baseline changes in activation, and that these patterns are changing significantly. They tend to change more significantly in premotor and posterior parietal regions. One way that we might be able to look at something similar with the current scan sequence that we've used in the study would be, instead of using kind of a monolithic mass that covers a representation of all 4 finger digits, we may want to kind of explore the localizer approaches like that Renzo used in your neuroimage work, where we basically can localize individual finger representations and maybe look at changes in patterns across those different masks. Another point to make is that, as I said earlier, so this again is from Jorn Diederiksen's group. And so typically with their fMRI work, they're looking at changes over much longer periods of training. And so maybe, some of these changes in fold or vasal response require much more extensive training before they become apparent with the current techniques available. And then, another point to make, this is from the same paper, is that we may be missing an effect because currently, we're focused on M1 as opposed to these premotor or posterior parietal regions. And so here, what they're showing is that you actually don't see much of a percent signal change in M1 with learning. And actually within the first week, you don't see it much at all when you compare trained versus untrained sequence activation patterns. And so again, this points to the fact that maybe we would want to, in a follow-up study, focus on premotor or posterior parietal cortex in regions that we know where these effects are emerging, and again, maybe over longer training durations.
Does your current sequence actually cover those areas by any chance, in addition to M1, or is it just covering M1?
Yeah, so I will go back to. So this figure here gives you an idea of where the current sequence covers. So we're seeing kind of information about the sequence encoded in addition to M1 in posterior parietal regions here, medial posterior parietal regions here, as well as mediotemporal regions in hippocampus. And so, yeah, these posterior parietal regions. And also we see DLPFC, or a transition area between ventral premotor cortex and DLPFC.
Are you asking about the vaso, the field of view?
Yeah.
Yeah. you do.
Oh, got it. Sorry.
So we use the same, like that Renzo and I both have used before, where it's a pretty small slab and we're trying to get it perpendicular to M1, to the hand knob, so they're thicker and different slice thicknesses, they're like double.
Yeah.
So, well, it kind of does. So we have
yeah.
premotor and more posterior parietal because the field of view is long, but
It's optimized for M1.
Yeah, it's perpendicular to M1, so you can get the layers really nicely there.
Yeah.
We might, in premotor, you can kind of see the ribbon. We haven't really tried to find layers there though, but it might be worth a shot.
Yeah, I think that's definitely a next step for the existing data set.
So I also have a question. It seems that just looking at the accumulated, like the percent change in one area, it seems that you're throwing away information of, if you try to take all the areas in that network, as many as you could capture in that slice, and just look at the connectivity strength as a function of layer, it seems like you would bootstrap a lot more power.
Yeah, I agree. It could be that because we're essentially reducing all of our data down to a single scalar for deep and superficial, And so, yeah, it's likely that, you know, because we're essentially optimizing for SNR, right? By doing that, essentially.
Well,
And
I mean,
so-
yeah, I mean, because it might be some idiosyncratic signal changes that might have a certain shape and timing, that all would be show this connectivity that would be unique in that regard.
Yeah. Yeah, so that's definitely something we should definitely pursue as the next step.
Yeah, we have a little bit, Peter. Like I said, you can see premotor cortex in our field of view, and we've defined it. We haven't tried to define layers there, but just like basically taking a univariate blob. And so then like trying to see differential connectivity to the layers of M1. We've been playing around with some of that stuff. Yeah.
We have a question in the chat from Farouk asking about the percent signal changes in VASO versus bold. Could you go back to the slide where you had the VASO and bold?
Yeah, I was comparing this with the Berlot paper that was shown in the last slide, because they had like 1.5% signal change in their study, like bold. But from the figures that I saw, and that's what I was asking for, like the percent signal changes you were seeing between rest and trial was like 0.0025 or something in VASO. Oh, it was 0.025, yeah, around that range. So I was just curious if this is like lower than expected, or is it within the expected range of VASO percent signal change?
So this is 0.025. Is this like percent then? It doesn't mean 2.5%?
Yeah, it's 2.5%. Yeah.
Okay.
Oh, then it's quite a lot. Like it's actually more than bold.
No, I believe that is probably converted to the percent. I think it is quite low.
Okay, yes, this is where
That's also
yeah, I apologize.
That's also a different score. That's practice minus rest. Yeah.
On adjacent practice and rest periods.
No, I was just curious, like if this is due to some design decision that is different in your study versus this Berlot study from Didriksen's lab. Did they have like longer rest periods? Did they have like stronger stimulation? I was just curious, like, because if it is 0.025% signal change, it's like really low, right?
Yeah, so I believe in the Didriksen work, I think the rest and practice periods were longer. I think they're typically using periods on the order of 30 seconds to a minute. So that could explain the difference.
Well, it's also bold, isn't it?
And also they're doing very different, yeah, so that's bold imaging. They're also doing very different designs where they're the participants are training on multiple sequences, and the sequences are structured in a way that allows them to get past the low temporal resolution to interrogate, you know, the sequence structure itself. And so they're training on these multiple sequence sets like over several weeks. And so, you know, you would expect to see a much larger, I think, difference in that case. Because remember, we're just looking at the initial training session.
Yeah, I mean, also like as the final thought with regards to my question. So if the percent signal change that you observe between VASO rest versus trial is like 0.025%.
Yeah.
Then like, I mean, you need to have a lot of data to kind of out that effect, right? So I am just now curious, like, is the effect extremely small? Did you have enough trials? And how much does it make sense to interpret this really minor percent signal change difference?
Yeah, the problem with adding more data, though, is that we're essentially just adding additional trials where they've kind of reached that plateau state in their performance. And then you're starting to introduce things like fatigue that could possibly impact the skill performance as well as the bowl response. And then you're getting into attentional factors, things like that. So it starts to get a bit murkier from the behavioral standpoint.
It's also where, you know, what he's showing there, our trial are block by block, so there's no averaging there of the
Yeah,
signal, right?
that's
Like, so,
right.
I mean, I can imagine if we did like more of a convention, because we wanted to see learning across the training session, I would imagine if we did more of a traditional, you know, average all your tapping versus your rest kind of analysis, that signal change would go up due to the averaging across the trials.
There's a lot of those questions could be answered by also looking at the bold, right? Because you have
Right,
it,
right,
and then the bold is more sensitive.
right.
And then, like the maybe main surprising thing is that, like, not only the lack of a layer distinction, which you kind of have, you see this difference in the superficial layers, but the overall scaling, there's almost no increase.
Right,
And in
right.
bold, then you would also not expect. Like, that's a low resolution question overall within M1. That's not a layer question. You don't need VASO for that.
Right, right, right.
So you could see if that's the same in bold and what kind of effect sizes you have there.
Right.
Yeah.
Yeah, I think pursuing these kind of like sanity check analyses with using the bold data, comparing it to previous work is important. Any other questions?
You brought up that, I guess this is just a general question, but you brought up that if you have more trials, you would be getting into the range where you'd start worrying about fatigue. Is there any reason to believe, so as you go through the different blocks, is it possible that people have fatigue effects as well? Or what, or if that's not a concern, why is that? Yes.
Yeah, so essentially what I'm talking about is if you carried this out further, right, you'd probably reach at some point where you'd start to see more variation in the performance or actual decrement in the performance. And so, you know, those would likely be driven by fatigue or tensional factors, as opposed to the underlying skill estimate, which is what we're really after.
I do have a curiosity question. Like, here you see the micro online is
Yeah.
negative, right? And also in Marlene's study, Bernd's group, you could see that you have this kind of micro offline jumps, but then within the blocks, it always decreases. How come that you get worse when you practice?
Yeah, so it doesn't always decrease online. And so it is negative here, slightly negative. And that's likely because our trial lengths were 20 seconds here. So you're more likely to get, you know, some decrement within each trial. So with the initial work, with the MEG and online crowdsourcing work, it's not negative. The micro online is, you know, it's pretty flat.
So it's not a slope of these little lines on the top.
So it is, yeah, so it is
On the top right,
if I
for
can-
example, on the
yeah, so this is the kind of this is the instantaneous performance within a trial, right? And what you see is, and exactly, like, these are actually the slopes. So the micro online would be the slope of this performance line here. And then the micro offline is essentially the slope or a line, the slope of a line that connects this last point with this first point, right?
and on the crowdsource study, on the like the figure on the right, it was always going down for the later ones.
Yeah, so what happens is that is that as you move closer to your performance ceiling, you're more likely going to go down.
Okay.
But what's interesting is that I'm not showing the data here, but even when you shorten to like 5-second trials, and so you're able to kind of extend this effect where you get these online increases and you extend them out further for more trials, you still see these like large micro offline gains. This is the reason why we kind of limit this analysis to this early learning period. So part of the initial paper, one analysis that was done was kind of looking at how this kind of inversion of the online curves impacts our micro offline analysis. So we did several kind of control analyses to address that. It's in the supplemental materials for the current biology paper. Yeah, my point is though, is that even when you manipulate that online part, you still see these micro offline gains.
Thanks. There are chat questions, and we're almost full by the hour. Sebastian, I think that was with respect to the previous discussion about signal changes. Maybe quickly, how is the baseline signal estimated? Right. Yes. So you showed the difference between. I read it out loud. Maybe I missed it, but what was the baseline to which the percent signal change was calculated?
It was an average across the 4 rest blocks. Here.
All right. Okay. So temporarily quite far apart from each other.
Yeah.
And there was drift correction for the entire thing.
Yes. Yes.
So it might. Okay.
We've thought about this too, and
Okay.
so like, should we average all 4 rests or just use like rest 1 for the training and rest 3 for the random, or
Right.
because the baseline estimates
Right.
could be weird, going across.
Okay, which could
entire
explain
exam session.
in the layer profiles, the 0 line was kind of halfway through, which could explain that.
Which one?
Like, yeah, you showed this average of over 20 people and the layer profiles with the 2 humps. There, the 0 line was kind of
High.
right, which could explain that. Like if you do a drift correction, then kind of your baseline varies across sliding globe.
Well, that's weirdly, I don't know why. So Rob made that figure. It's Z-scored there instead of
Okay, so it's
percent
mean-
signal change.
mean.
Yeah.
But still, like
But-
the underlying record, if the micro offline learning happens, it happens in the rest block, right, to me, so.
So like you mean like the rest block after
Yeah,
the
not just
training?
between the
Yeah, but
rest
that,
block,
like-
But also in the rest of blocks, longer rest blocks.
Totally, yeah.
Oh,
That's what we
yeah.
talked about.
So in the MEG work, what we found is with respect to the replay density, you see a pretty quick state change. So once they're in these longer rest blocks, they're not showing these replay events at a high rate anymore. So it's like they're outside of their kind of task engagement state.
Yeah, because in the intro you mentioned something about sleep, so I was thinking
Right,
in
right.
that state it was similar like this rest 1, 2, 3, 4, but that's where you see the highest micro offline gain, if I remember correctly.
Right, so you're talking about here, right?
Yeah.
This
Right,
retest, yeah. So actually, the change here is not as large. So this is about what? It's like 4.5 sequences per second. So what we're talking about is these micro offline gains here account for more performance gains than these overnight offline performance, because all of the micro offline gain increases are accounting for this part of the learning curve in the kind of training session, right? And that's like, in this case, that's about like 8 sequences, right? And here you get about a 4.5 sequence increase.
Well, that huge increase too, I mean, it's totally due to the sleep, right? Isn't that the point? Like, if there's 12 hours intervening with no sleep versus the 12 hours.
Yeah.
Is that what you're looking at?
Yeah, I think I probably misunderstood this figure.
Yeah.
The point still is a good one, though, about like the rests that are right after the trainings, and there could be residual effects or.
Yes, I wonder if you just change to the rest 1 for the first block and then the rest 3, would that change the baseline hugely?
Yeah,
That's kind of an empirical question.
totally, and we have somewhere like.
And so this is,
Like,
and just one more point, this is what we found in the MEG study for the replay density. So you get a quick shift back down once you're out of the practice block.
Oh,
Yeah, but it also still makes sense that the fMRI and the MEG are different kind of signals. So
Yes.
it's worth checking out what happens when you separate the baseline from more local baseline.
Yep.
yeah, we've been looking into that, like we have, all this stuff.
Okay, last question. Boris has a question in the chat. Do you want to unmute and say it yourself, or I can read it out loud? There are slow fluctuations in error rates across time during block. Maybe this refers to the burn stroke
Here.
Again, I'm not sure. Is this related to arousal level? Did you measure pupillary responses?
We did not do pupillometry for this data set. So we do actually have these data for our MEG study. And I'm just trying to remember how it relates to errors. So you definitely, when the subject, so there's no error feedback given in this task. All they're given is feedback about where they are in the sequence. They do recognize errors though, when they make them. And we're actually able to predict when an error will occur. We have another paper that came out recently, where we can predict based upon the key press transition times, as well as MEG activity features, like when an error will occur, and also with the pupillometry data, we do see an increase in pupil diameter that's local to an error event. In this case, I will say that one difference between the MRR environment and the MEG environment is that we do see slightly higher error rate. It's still, it's in the range that we'd expect. So, it peaked here around 15%, but normally it was, on most trials, it was between 5 and 10% errors. And that was for the training data set. This is what we expect to see, somewhere around 5%, where it peaks out at after the sequence stabilizes. So yeah, it's possible that the error is going to contribute in some way to the vasal response we're measuring, but we haven't looked at that in detail. Yeah.